The human immune system is aimed to encounter and eliminate invading pathogens while being harmless to our own body. When these self-limited, tightly regulated responses are impaired, a damaging assault against the body's own tissues may arise and result in an immune chronic inflammatory disease. A few of the most prominent chronic inflammatory diseases are psoriasis, multiple sclerosis, rheumatoid arthritis, inflammatory bowel disease, and atherosclerosis. VBL pioneered the lesinoxoid class of rationally designed oral anti-inflammatory agents to address the substantial unmet needs in these immune inflammatory diseases. During the inflammatory process, T cells interact with antigen-presenting cells called dendritic cells and macrophages. This interaction induces the secretion of the cytokines IL-12 and IL-23 from the antigen-presenting cells, leading to T cell activation and pro-inflammatory cytokine excretion by the T cells. Consequently, T cells acquire the ability to migrate from the bloodstream through blood vessels to the inflamed tissue. Once infiltrated, T cells are reactivated and secrete the pro-inflammatory cytokines interferon gamma and IL-17. Concurrently, monocytes are being recruited by locally secreted chemokines and penetrate into the tissue to take part in the inflammatory response. This convergent activity augments inflammation in the affected tissue. VBL is pioneering a new class of drugs called lesinoxoids to treat immunoinflammatory conditions. VBL's lead compound, VB201, is an orally available, rationally designed phospholipid analog from the lesinoxoid family. VB201 has significant potential to treat inflammation in chronic diseases such as psoriasis, rheumatoid arthritis, multiple sclerosis, inflammatory bowel disease, and atherosclerosis. VB201 inhibits the production of IL-12 and IL-23 by dendritic cells and macrophages and acts as a counterbalance to the pro-inflammatory immune system activity. VB201 has a highly specific and targeted activity that does not affect system-wide immune factors. As a result of VB201 activity, inflammatory cells such as T cells and monocytes are impaired in their ability to migrate through the blood vessels and infiltrate into the inflamed tissue. Moreover, T cells that were able to migrate will secrete a reduced amount of pro-inflammatory cytokines such as interferon gamma and IL-17. Inflammation is now restrained. VBL Therapeutics is committed to developing first-in-class safe treatments for chronic inflammatory diseases. VBL's novel drug has the potential to improve the quality of life and the life expectancy of millions of patients around the world.